Dr. Lorianne Masuoka della Marinus Pharmaceuticals, che ci illustra i risultati della fase 2 di uno studio in aperto con il Ganaxolone.
Thank you very much for inviting me to give this talk. I'm very pleased to present the results of our ongoing trial of Ganaxolone in children with PCDH19. This is a program that's been going on for a couple of years, and we've recently announced results in a different population of CDKL5 deficiency disorder, and we are very pleased to announce that we will be proceeding with the first phase 3 study in these children, so that's moving ahead very well. So just to give you a little bit of background as to what is ganaxolone, Ganaxolone is a specially engineered form of allopregnanolone, which you've just heard about very nicely from the last speaker. So allopregnanolone is a neurosteroid that's produced in the brain, and it is a very important neurohormone that is responsible for quieting the brain and keeping it from being in a hyperexcitable state. And without adequate levels of allopregnanolone, as you've learned, there are problems with seizures and learning abilities and anxiety and that sort of thing. Ganaxolone's been studied in quite a large number of people. It is known to be a very potent and long-term positive modulator of GABA-A systems. We currently have it available in intravenous forms. We have it in an oral suspension form for young children, and we also have it in capsule forms for adults. And we are currently studying it in multiple pediatric epilepsy indications. And right now we are continuing in CDKL5 deficiency disorder. We are in PCDH19 epilepsy. We have patients in long-term follow-up, and we are considering what to do next in PCDH19 epilepsy. And to be honest with you, part of the reason why I'm in this conference is I want to show you the data, to tell you what progress we've made and what we've learned, and to ask you your opinion as to what you think we should do next. I mean, that's honestly what we're here, is to find out what your opinion is. I was here in Rome in April at the CDKL5 conference, and on the basis of that Congress, we put a lot of research into it, and we raised a lot of money, and we are going into CDKL5 in a very big way, and that's part of why I'm here, is to get your opinion, because it matters. We're also studying Lennox-Gastaut syndrome, and we will be presenting an abstract at AES on those results. So ganaxolone targets exactly the same areas of the GABA-A receptors as allopregnanolone. So it binds to both synaptic as well as extrasynaptic areas of the GABA-A receptor. Now, what's important about that is it does not bind to the benzodiazepine receptors. And these extrasynaptic receptors are particularly important because they mediate tonic inhibition. So these are things that are constantly present. They do not become internalized. And it means that you can keep giving the drug without having to give higher and higher doses, and you still get the same amount of effect. And what I can tell you is that in our study of 30 subjects, 30 patients in our pediatric epilepsy study, 10 of those patients are still on therapy for over 2 years. And so these patients are able to take the same exact amount of drug without having to take holidays or having to stop their drug like you see with benzodiazepines. So what are the results? First of all, the study design, it's an open label study that's being performed in the United States and one site in Italy at the hospital Bambino Gesù, where Dr. Marina Trevisano is one of the investigators along with Dr. Nicola Specchio. We are evaluating 3 cohorts of patients with epilepsy, as I mentioned, PCDH19, CDKL5 disorder, and LGS. We have enrolled 11 female children with PCDH19 epilepsy, and they ranged in age from 5 to 16 years old. And this included a 12-week baseline period where they had 4 weeks of recorded seizures in their diaries, as well as 8 weeks of recorded diaries that they had from before the time they came into the study. They were then treated for 26 weeks, followed by the option to go into a 52-week extension. So what I mean by that is that if after 26 weeks they had at least a 35% reduction in seizures, they were qualified to go on for an additional 1 year of therapy. So they had to have good seizure control. And coming into the trial, they had to have at least 4 seizures in a 28-day period, so they could not be seizure-free. The primary efficacy endpoint was the change in seizure frequency over a 28-day period over the 26 weeks. So over a 6-month period of time, they had to have, we looked at what their monthly change was in seizure rate, but we also looked at their change in seizure-free days. And I will say that most of the patients that came into our trial with PCDH19 had mostly seizure-free days. Their seizure-free day rate was about 20 coming into the study. So as many of you know, these patients had lots of seizures, but they were mostly clustered. So they were clustered over maybe 10 days of time. Most of their time was seizure-free. So this is not meant to be read. It's just simply to show you a chart that we titrate the patients slowly over a period of about 2 weeks where they get increasing doses, and it's based on size. So if the child is small, they get oral suspensions in syringes, and if they're larger, they can take capsules. But for those kids that have trouble swallowing, they can always take the liquid suspension. And it's often given with meals or with milk because it's better absorbed. Now these are the results. What you can see in blue is that we had 4 subjects that are considered responders because over the first 3 months they had a greater than 50% response rate. And we had 1 subject that had a 100% reduction in seizures, but unfortunately developed a rash and was not rechallenged. It's unclear whether the rash was due to the seizure because the patient also had a febrile illness, had a high fever, was very ill, and had to be hospitalized, so it was unclear if it was due to a virus. But just to be safe, the patient was taken off of the medication and not rechallenged. But she had a very good response. Another patient had an almost 80% reduction in seizures, but towards the end of the 6-month period, the seizures had returned. And one patient who had a reduction of 74% went on to the 1-year extension and is now seizure-free and has been seizure-free for now almost 2 years. I put 2 patients into yellow because they had about a 1/3 to 1/4 seizure reduction. And so the question I'm going to be asking you at the end of this presentation is whether or not that's good enough, whether you would want to be on a drug that had a 1/3 seizure reduction. And then the one that I have that is in question mark is patient number 8. This is a patient who had a very almost meaningless reduction in seizures. This was off her medication because the doctor said, this is a seizure trial, and if your seizures aren't getting better, you have to come off. And the mother begged the company and begged the doctor to please keep her on the drug because she was able to go to school for the first time in her life, because not only was her behavior better, but her cognition was so much better. And we had to say that we have to go along with the doctor. It's the doctor's decision. And she made many appeals, and then finally she was taken off the drug and she was no longer able to go to school. She had to drop out. So that was a very sad case. We still stay in touch with the mother, and her hope is that as the drug becomes approved, her daughter can go back onto the drug. So one of the questions I'll be asking you later at the end of this presentation is whether we should be studying autistic behavior and educational abilities and cognition in addition to seizures, so that patients can stay on drug if they show benefit in those areas as well. So this was our first study in PCDH19, and we're still learning. So how do we identify the best responders to ganaxolone? One thing I should note is that, for example, in this patient here, let's see if I can this patient would be considered a non-responder. But here you can see the patient had a 25% response rate at the 6-month point. Now that's cumulative. So what that means is that they did not have a decrease in seizure rate in the first 3 months, but by the second 3 months, the seizure reduction rate was more like 50 or 60%. So this patient would be considered a non-responder, but their response to seizure medication was going higher and higher and higher. And the doctor actually called me and said, could you make an exception? Because this patient is only at 25% and your cutoff rate is 35%, and this was a very famous doctor in New York. And he said, the parent is telling us that the seizures are not only less frequent, but they're much less severe, and they are shorter duration. So I made an exception, and we let that patient stay on. And by the first 1 month in the 52-week extension, she would have formally made their criteria because the seizures kept getting less and less. So that was a case where we made an exception, and we made the right decision because the seizures kept getting better and better. So this was an unusual case where there was a late responder. Normally, when they have a response, the response occurs early. So should we consider allowing patients to stay on drug if they have a late response as compared to an early response? Should we be measuring seizure-free days, or should we just be looking at reduced seizures, which is the typical way to look at it? In other words, what is the better predictor of quality of life? You know, what's important to you, the parents? What's important to you, the doctors? If somebody has a rash, should we give them some steroids or Benadryl, and should we re-challenge them, especially if they have a 100% reduction in seizures? You know, would that be worth it to you? Would it be worth the risk? Especially if you're not sure about the rash. I will say, we've had several other children with minor rashes. We re-challenge them and the rash doesn't come back. I know personally I had a rash with penicillin, and I had to take penicillin again, and I didn't have the rash again, so sometimes that does happen. Should we be measuring pharmacodynamic measures, biomarkers such as hormone and steroid levels? I will tell you that at this time, if I'm invited back, if my talk is good enough and you invite me back next year, I will be able to tell you that we are getting information about why those patients in blue, that I can't go back up to because I can't find the keyboard, why those patients in blue are doing so well and why the patients down at the bottom are doing so poorly. They are different. They are different in terms of their blood levels of certain chemicals. In terms of adverse events, they're quite benign. There are children who temporarily get tired when their doses go up, but they get used to it. The somnolence or the fatigue goes away. They report some headache and fatigue, but in general, the side effects are very mild. And compared to most anti-epileptic drugs, this is considered to be a very well-tolerated drug compared to the vast majority. I will say in the CDKL5 population, we've had 0 complaints of somnolence or fatigue. So the lower, it seems like the lower the cognition, the better tolerated this product is. So, you know, really it just shows that the patients that do well are a bit more sensitive. You know, first of all, for the families and for the doctors, please participate in natural history studies. It's enough, it is a good start to establish a registry, but please participate in registries that incorporate a high-quality natural history study. It's very, very helpful to know what is the percentage of patients that have A, B, C, or D, because we will incorporate that into our clinical studies. If autistic features are a big part of what you have, then we will put that into our clinical trials and measure it. But we need to understand how that progresses over time. So for example, if we have a 1-year follow-up study, we need to know, is that natural history or is that our drug? And the FDA, for example, will allow us to run single-arm studies where you don't have to have a placebo if you have a good natural history study to use as your control. So if you establish a natural history study, we might be able to get rid of placebo-controlled trials. Otherwise, we have to do a placebo-controlled trial. We want you to provide feedback about what a successful drug trial looks to you. We just heard just now that there are no good results from allopregnanolone and engineered allopregnanolone studies. I believe that was my study that was mentioned. So the question is, are these results good enough to continue going? If the answer is no, we won't continue to go on. We'll move on to other things because let me tell you, these trials are very, very expensive. This trial alone was probably about $15 million. So we won't continue if you think it's not good. And would you put your child or your patient into another trial? Because if you wouldn't, then it's not worth our continuing. And but the one thing I will say is, whether you participate in a ganaxolone trial or not, please do participate in some clinical trial, because without clinical trials, there will be no new drug options. So that's it for me. Thank you very much.
Thank you very much. The last message is very important. Now, there are some other experience. Prima le signore, quindi...
Sì, grazie. Da quello che so io, il primo trial col Ganaxolone non ha previsto le misurazioni di allopregnanolone sulle pazienti prima e dopo l'assunzione del farmaco. Ora, se questo è corretto, secondo me, insomma, anche da non tecnico, un eventuale ulteriore trial sarebbe necessario invece introdurre questo elemento, perché questo ci consentirebbe anche di comprendere se c'è effettivamente una correlazione tra questi bassi livelli di allopregnanolone che sembrano avere le bambine e la patologia. Quindi evidentemente laddove il Ganaxolone portasse a dei risultati soddisfacenti e contemporaneamente ad un aumento di allopregnanolone, sarebbe un'informazione importantissima e per il trial, ma anche per tutto quel filone di studio che riguarda, insomma, il coinvolgimento dei neurosteroidi nella malattia. Grazie.
That's a very good question. I will be honest with you, I am not familiar with a clinical trial of allopregnanolone in children with PCDH19. It was my understanding that the Ganaxolone trial was one of the first trials in children with PCDH19. But I will say that in our trial, we collected throughout the entire trial at baseline, as well as at many time points, we collected about 12 neurosteroids, of which both allopregnanolone and allopregnanolone sulfate were 2 of the 12.
Quindi li avete misurati?
Yes.
E avete anche misurato dopo il trial, avete visto, quindi è possibile sapere che se ci sono stati dei cambiamenti, insomma, nei livelli di neurosteroidi nelle bambine che hanno partecipato al trial?
So we studied the neurosteroid levels throughout the trial, which includes through, I would say, a period of almost 2 years. We do not have data after they exit the trial, so we don't have data when they're off the drug, only data when they're on the drug. So we have multiple time points, so we take neurosteroids at every visit.
Va bene, grazie.
Più un commento che una domanda. Come endocrinologo non credo che vi sia un razionale che giustifichi, diciamo, la somministrazione di ganaxolone per aumentare l'allopregnanolone. Cioè il ganaxolone sostituisce l'allopregnanolone che manca. Penso che il razionale possa essere questo. Ecco. Se, diciamo, si vuole aumentare l'allopregnanolone bisogna utilizzare qualcosa che ne stimoli la produzione. Nel nostro caso, ad esempio, l'ormone adrenocorticotropo è, diciamo, un ormone che può aumentare la produzione di allopregnanolone. Sì, o il progesterone, che è un precursore, può aumentare la produzione di allopregnanolone, ad esempio. Ganaxolone non penso proprio che possa avere questo tipo di...
So, I'm sorry, the English sort of cut out. So are you saying that we should not pursue ganaxolone in PCDH19 Epilepsy? I'm sorry. Yeah, so ganaxolone is a form of allopregnanolone. The reason why it's engineered is so that it cannot convert to progesterone. It doesn't interact with the enzyme that converts allopregnanolone back to dihydroprogesterone, to 5-alpha dihydroxyprogesterone. So that's the reason for the engineering. But it is, in fact, it does bind exactly the same way allopregnanolone does. So originally the reason why, it's actually Dr. Getz, the reason why Marinus pursued this product in PCDH19 epilepsy was the idea that if they're deficient in allopregnanolone, that perhaps providing them with extra allopregnanolone might actually help with the seizure disorder.
If I'm correct, I saw you showed in one of your first slides that the duration of the trial was 26 weeks.
The main study was 26 weeks, and if the patients were doing very well, we felt for ethical reasons we had to offer them extended therapy. We actually thought very few people would go on to extended therapy. We were quite surprised that quite a few did.
Well, now this is positively long. Because trials for seizure medication are usually too short to understand
Yes.
their effect. Was that something you have chosen for specific reasons, that 26 weeks, or was imposed by regulatory indications?
So the regulatory authorities are very accepting of 12-week trials, whether you like that or not. And it sounds like you don't like that. They will approve drugs on the basis of 12-week trials. The reason why we wanted to do a 26-week trial is that this was an exploratory trial, and we wanted to see what the effect of the drug was over a long period of time. And we got more than we bargained for, because we now know what the effect of the drug is over 1.5 years to 2 years. And what we're finding is that if you have a response that's large, like a 75%, 80%, 90% reduction in seizures, especially early on, that response tends to be durable for a very, very long time. It tends not to go away. If there's no response in the first 6 months, there tends not to be a response thereafter. So the kids then drop out of the trial at 6 months. So it's funny. And I think we're starting to figure out why. But it appears that you're either a responder or you're not.
Yeah, but this is very positive in itself, because if you consider one of the features, the fact, for example, of having cluster of seizures during fever and so on, there are other situations like Dravet syndrome where trials with other medications have been lasting for maximum of 2 months. So during the 2 months periods, a typical patient might not experience the most stressful periods with fever and other precipitating factors, while if the trial is long enough, 26 weeks, that will certainly repeat real life.
Yeah.
So it's a very positive aspect.
Yeah, when you go 6 months, if you have a honeymoon period of 2 months, it'll get washed out, so that by 6 months you don't have significance. So we have a very high responder rate at 6 months. And so it means that anybody with just a honeymoon period would be considered a non-responder.
Sorry, I have a quite different question. In your first slide, you shown that your experience is also in CDKL5 epilepsy, okay? I don't know if it's possible politically for you, but it's possible to have an idea of the possible differences in the answer in the two different populations? Considering that in CDKL5 epilepsy, there is no evidence of a similar hormonal disorder.
So I would say, number one, stay tuned. We don't know that. We are collecting and have collected years of neurosteroid levels in CDKL5 patients, in Lennox-Gastaut patients, in Fragile X patients, and in patients with focal onset seizures. And until we started to really dig deeply into the PCDH19 data, we haven't really looked at those data carefully. And we are now on almost a religious mission to look into these data. And so in a year from now, we'll have a lot of information to answer that. The clinical impact on CDKL5 is quite different in the sense that this drug works very well on generalized tonic-clonic seizures and on drop seizures. And these are the typical seizures that happen to CDKL5 disorder patients. So the effect on CDKL5 patients is quite dramatic. And the impact on seizure-free days, we have an increase in seizure-free days in the CDKL5 population of 80%. So we're making them almost completely seizure-free. And so the CDKL5 patient population has, as you probably know, on average about 10 seizures every day. Their seizure-free period coming into the trial was about 1 to 2 days on average, as compared to 20 days for the PCDH19 patients. So, the effect in CDKL5 is very obvious. But whether or not it's working because of neurosteroid chemistry, we can't prove it yet, but I suspect there's probably something there. And as soon as we're finished studying the neurosteroid levels, we'll let you know.
Thank you.
And thank you for asking in English.
Through Dr. Specchio, I've been asking longer than 3 years to include my daughter into Ganaxolone trials, but I was refused because when the trial started, she was already 18 plus, and the Ganaxolone trials was only for girls,
15.
yes, till 11 years old. No?
15.
Yes. But first of all, my daughter is very sensitive to steroids, and sensitiveness to steroids increase after puberty and not before puberty. So now she's been taking high quantities of cortisons for longer than 10 years, and side effects are already destroying her because cortisone cause osteoporosis and many other problems. No? So I wonder then Ganaxolone could be a good replacement for cortisone. And also I wonder that the hormones play an important role in epilepsy after puberty and not before puberty. So I hope that now is a time that also my daughter could be included in Ganaxolone trial.
Yes, thanks for that question. We are in the process of designing our phase 3 trial in CDKL5 disorder, and we will be extending the age range and looking more into post-pubertal children, predominantly female. CDKL5 has more males than PCDH19, but still mostly female, so we will be looking at that. You know, what's really lacking is natural history as to whether or not seizure types and things change with puberty. That would really help us to know what age ranges we should study. That's why I say these natural history studies are so important. Now, whether we would include a higher age range in PCDH19 epilepsy has a lot to do with whether we would do another study in PCDH19. And again, that's one of the reasons why I'm here, because I have heard rumors that the PCDH19 community finds the results that we've gotten in PCDH19 to not be that great. And if that's true, and if you're looking for something better, then we wouldn't do another study. So it really, one of the reasons why I'm here is to know that information directly. I will say in the CDKL5 patient, we do have a patient who came in just on steroids, and she was having 10 seizures a day, and she's now seizure-free, and they're going to take her off her steroids because her steroids cause side effects. And so it is possible that ganaxolone could work well when steroids are not working so well. Her steroids used to work. It's only an N of 1, and it's not PCDH19. So I would say, especially with behavioral issues, it is a possibility. But again, the only way to know would be to test it.
Sì, vorrei fare solo una considerazione in generale che forse non è strettamente legata al ganaxolone, che premetto la mia bambina ha testato, insomma ha fatto il trial qui a Roma, il Bambin Gesù, ma forse in generale a tutti gli antiepilettici, che sono purtroppo quelli che sono gli effetti collaterali che per esempio nella mia bambina si sono dimostrati molto evidenti. C'è stata una riduzione non molto evidente delle crisi, però ha sviluppato un'aggressività poco gestibile. E volevo solo far presente che molto spesso noi che abbiamo già delle situazioni molto difficili nel gestire l'epilessia e quindi le crisi, i cluster che si presentano, facciamo fatica poi a gestire anche questi aspetti collaterali che molto spesso sono forti e violenti. Quindi se posso dare un consiglio è di lavorare, se possibile, anche su questi aspetti qua.
So, yeah, so it is true that every drug comes with a side effect, and the question really is, for those kids that have side effects, they have to stop, and for those children who have major improvement and improvement in their autistic features, they continue. There's, I think, no scenario in which the drug is going to be good or perfect for everyone. And what we're now trying to look into is, why is it that some kids, and I showed you in the bottom 3, that there were kids who, perhaps their behavior was worse. I suspect that that child might have been in the category where the seizures were also worse, clearly they were worse after taking ganaxolone. And so the question is, how do we distinguish even before the child takes the ganaxolone whether they're likely to be worse versus the ones at the top where they've done so well, kids going to school, their behavior is better, they're less autistic. How do we discriminate between those two groups even before they take the drug so that it doesn't become such a blind experiment? Because wouldn't that be great? Because with every other anti-epileptic drug, that is exactly what it is in the case of each child. It's an experiment. You don't really know ahead of time whether or not you're going to have bad side effects or a good response. You have to just try. So we're hoping that we can develop something where we can already predict a little bit.
Since this study is very expensive, as you said, and before really throwing away the ganaxolone that has the potential to be a good drug, I was wondering if the type of study was actually the good one. I mean, you said the study was an open label study. I just wonder whether we should, maybe a different type of study could be more effective to show that ganaxolone is an efficient drug or not, perhaps comparing with with the other medications. I don't know how the study was designed and why you picked this kind of study design. If they, of course, certainly the pharmaceutical company has taken advices from specialists in this, but I was just curious of why did you decide to do this type of study?
Yeah, this was the first ever study in pediatric epilepsies, and it was only supposed to be a few kids with PCDH19. epilepsy. And so it was meant to be a safety study, just to see if it was safe to treat children with genetic epilepsies. So it turned out that there was some kids that just did so well, it just took on a life of its own. And as PCDH19 kids, some of them were doing extremely well. Somebody said, well, why don't you study CDKL5 patients, because they seem to have a disordered GABA system. And then someone said, well, why don't you study Lennox-Gastaut syndrome? And then, it sort of took off from there. It was never meant to be such a big study. The next CDKL5 study will actually be a controlled study, and if we were to do a PCDH19 study, it would be a controlled study.
Yeah, that's what I would suggest. I mean, I would say the controlled study, probably double-blind, should be the way to go, because that's really,
absolutely. Yeah. No, in this first study, it was meant to just be a safety and tolerability study.
Ok, l'ultima perché se no...
Very quick. Talking about registries, obviously as parents, we are very glad to participate in registries, but instead of create another new registry that will be only valid locally, would you suggest us a registry that we should get involved with, we should participate? Today we heard about the one of University of Adelaide. Will it be a good one for you to, would you suggest that? Would you partner with that?
Yeah, I mean, I think we'd be willing to partner with any registry that has a natural history component. In the United States, there's one by NORD FDA. So the FDA has collaborated with NORD to set up natural history databases. But what's available here in Europe, we collaborate on the CDKL5 front with 2 different registries. So we're not here to choose what's the best. We look to you, the parents and the patient advocacy groups, to tell us what's the best. And we're willing to collaborate with anybody who is willing to provide these data. We're willing to help you, to get the data and do whatever we can to help you to get these databases going.